Hello, it's my pleasure to welcome you to the first in a series of virtual scientific sessions developed by the American Neurogastroenterology and Motility Society, focusing on basic concepts in neurogastroenterology. My name is Beverly Greenwood van Meerveld, and I'm the immediate past president of the ANMS and the current director of the ANMS Institute. The goal of these virtual basic symposia are twofold. First, to provide you with the latest information on a basic science topic related to our field of neurogastroenterology and motility, and also to provide you with a positive distraction from the pandemic and allow us to connect virtually with leaders in our field of neurogastroenterology and motility. The topic of today's symposium is the microbiome, the role of the brain-gut axis, and we will have 3 amazing presentations from experts on this very exciting topic. But before we start, I'd personally like to thank each of our 3 speakers today for taking the time out of their busy schedules to join us. I would also like to thank members of the ANMS Meetings Task Force for developing this exciting virtual program that over upcoming months will cover multiple basic concepts in neurogastroenterology and motility. It's now my pleasure to introduce today's moderator, Dr. Purna Kashyap, who is an associate professor of medicine and of physiology at the Mayo Clinic in Rochester, Minnesota. He's also the past ANMS counselor and has played an instrumental role in developing the program today. Thank you, Purna.
Thank you, Beverly. Thank you for the kind introduction. And I am really excited to start this session on microbiome and the gut-brain axis. Nothing better to discuss at the end of the day to invigorate ourselves. As a quick reminder, we'll do Q&A at the end of all 3 talks. Please don't use the chat function if you want to send questions, but submit your questions to the Q&A. With that out of the way, it is my pleasure to introduce our first speaker of the evening, Dr. David Reed. Dr. Reed is an assistant professor and clinician scientist in the Department of Medicine and the GI Diseases Research Unit at Queen's University in Ontario, Canada. His research focuses on how the interaction of intestinal luminal factors like the dietary components and stress or the microbiota modulate pain signaling and motility in disorders such as IBS. He's also the past recipient of a research grant from ANMS. He will tell us today if the gut microbiota should be blamed for abdominal pain. Welcome, Dr. Reed.
All right. Thank you, Purna, for the introduction and for the opportunity to speak today. And as you mentioned, the topic I was given is, are the gut microbiota to be blamed for abdominal pain? And I think to get to the answer right at the beginning, I think it's likely, and I think it's in some people, and even more so, to be accurate, it's in certain situations or specific circumstances. I'll see if I can sort of convey that in the next 15 minutes or so. So this is a schematic demonstrating that we've learned that they're both peripheral and central factors that influence visceral pain. And we've learned over the last few decades, there's a number of host mediators that are secreted by various cell types that can influence visceral pain within the gut. But of course, what's missing from this slide is the microbiota, which we know there's a lot of microbiota host crosstalk in various GI disorders. And so one of the first questions that one can ask is, can the microbiota modulate host-derived mediators that sensitize pain pathways? One of the simple ones to look at is serotonin, or 5-HT. As we all know, it's well recognized that serotonin is an important regulator of gut function, and it can increase the activity of nociceptive nerves, which I've shown here in this schematic, which shows an enterochromaffin cell, which houses a lot of the serotonin within the gut, which when stimulated can release the serotonin on the basolateral side, which in turn can activate 5-HT3 receptors on nociceptive nerves whose cell body reside in the dorsal root ganglion, or DRGs. And then this information is conveyed on towards the brain. So does the microbiota, for example, modulate 5-HT within the gut? Well, in fact, our moderator, Dr. Kashyap's group, has looked at this, and what they did is they colonized germ-free mice with human fecal microbiota and measured colonic 5-HT levels and found that the humanized gnotobiotic mice had increased 5-HT within the colon compared to the germ-free mice, which is shown in white. And the human gnotobiotic mice actually, although it was less than the conventional mice, it was not significant difference between the 5-HT levels. They then went on to look at pathways involved. It could show similarly that in the human gnotobiotic mice, they had increased amounts of TPH1 protein, which is the rate-limiting enzyme in the enterochromaffin cell in the production of serotonin in those human gnotobiotic mice compared to germ-free mice, and were very similar to the conventional mice. And then they further went on and showed that it appeared that short-chain fatty acids were also involved in this pathway, which of course are produced by bacterial fermentation within the gut. But moving beyond just mediators that can be, the microbiota can influence, can the microbiota actually increase pain? And this is a study in which they transplanted the microbiota from irritable bowel syndrome, or IBS, constipation predominant patients into gnotobiotic rats, and then measured the visceromotor reflex. And to remind you, the VMR is performed when you insert a balloon into the distal colon of a rodent, distend that balloon to different volumes or different pressures, and then measure the number of abdominal contractions, with the increased number of contractions analogous to increased pain. And in this graph here, you can see that in the rats that were colonized with the IBS fecal microbiota, there was an increase in the number of abdominal contractions, or an increase in the VMR in these rats, as shown in the black diamonds, compared to mice that were colonized with healthy microbiota, shown in the black squares, or the conventional rats, suggesting that the microbiota may in fact influence pain. Well, what about the mediators that might be secreted by the microbiota? And this was looked at a number of years ago by Lionel Bueno's group, in which they took fecal supernatants from IBS diarrhea predominant patients and infused it into mice, and an hour later looked at the VMR response. And you can see in this graph that the IBS fecal supernatant, which is the black squares in this graph, increased the VMR in mice compared to the control mice, which are shown in the black circles. They then went and incubated that fecal supernatant with serine protease inhibitors and infused that into the mice, and found that that reduced the VMR response in the mice, suggesting that the protease was involved. They then went on to look at the receptors, and they used the protease activated receptor 2 deficient mice and infused the IBS fecal supernatant in these mice, and also found that it inhibited the VMR response compared to the IBS fecal supernatant infused into the wild type mice. And this all suggests that the microbiota in these IBS patients is secreting a pro-nociceptive protease. When we look at a lot of the evidence that's been generated in regards to the microbiota modulating pain, most of the studies in the past have been in vivo, either in germ-free mice, which I've shown you, or antibiotic treatment early in life, or even using single strains of probiotic bacteria. But there's been a shortage of evidence of direct microbe to neuron communication. And this is shown schematically in this slide, in which the microbiota in the lumen can secrete mediators which may impact the barrier function, so increased permeability, allowing various mediators such as bile acids across the epithelium, which can increase nociceptive nerve activity. And these microbes may also be communicating with immune cells or enteroendocrine cells, which can release mediators and subsequently modulate pain. But there's been not a lot of work until recently that has asked, do the microbes secrete mediators that directly influence nociceptive nerve activity? And this has been looked at more recently by a colleague here at Gidru at Queen's, Dr. Alan Lomax. And what he did in his lab, they used MET1, or Microbial Ecosystem Therapeutic 1, which is actually a simplified microbiota of 33 strains of bacteria, from a healthy individual. And this MET1 was created in a collaboration between Dr. Elaine Petroff here at Queen's and Dr. M. Alan Vercoe at the University of Guelph. And it's been shown that the MET1 is been used as a synthetic stool for transplant with promising results for recurrent C. difficile infection. But what the Lomax lab did is they took the supernatant generated by that microbiota and incubated it with DRG neurons, and then measured the excitability of these neurons. And to do so, they performed patch clamp recordings and measured the rheobase. And the rheobase is the minimum amount of current you need to inject into a neuron to generate an action potential. And so a higher rheobase would be analogous to a lower excitability of the neurons. And you can see in this graph that the MET1 supernatant at a dilution of 1:100 increased the rheobase, suggesting that the supernatant was decreasing the excitability of these neurons. We collaborated with Alan's lab and performed some colonic afferent nerve recordings and could show that the MET1 supernatant also decreased the spontaneous firing of these afferent nerves within the colon. Jessica Sessamine, who was a PhD student at the time, performed majority of these experiments, then looked at the mediator secreted by the MET1 and found that when she used a serine protease inhibitor, FUT175, which did nothing to the excitability when given alone to control neurons, significantly reduced the effect of the MET1 supernatant. She then went on to look at the receptor that might be involved. And when she used a PAR4 antagonist, which also did nothing to the excitability of the neuron when given to control neurons, significantly inhibited the effect of the MET1 supernatant, all suggesting that the microbiota, in this case a commensal microbiota secreting an anti-nociceptor protease that's working through PAR4 on DRG neurons. Well, what about down at the level of the gut? And this is some recent work that we've been doing, and this has been done by a PhD student, Amal Abdu Omar, in which she made ex vivo recordings of these colonic afferent nerves. And so this is a schematic showing that she would take the distal colon and she would cannulate it so it could perfuse Krebs or fecal supernatant through the lumen. And then you can also close off the outflow to allow the colon to be distended up to a pressure of 60 millimeters of mercury, all while recording the activity of the nerve. And in the bottom left is an example of one of her experiments in which the top panel is the raw trace of the nerve activity. This blue bar is when she distended the colon, and then the bottom panel in pink is the rate histogram showing the firing frequency of the nerve. And you can see it spontaneously firing, and then when she distended the colon, there's increased activity of the nerve as indicated in the rate histogram. She then superfused the fecal supernatant from healthy volunteers, and not only did she see a decreased firing here in the spontaneous activity, but the response to colonic distension was also reduced following the healthy fecal supernatant. And this is then summarized in the right graph, showing that the healthy fecal supernatant actually decreases the mechanosensitivity of these colonic afferent, and these are high threshold units, which we think are the nociceptive nerves within the colon. This is contrasted when she then looked at IBS diarrhea predominant fecal supernatant. Again, down in the left side, you can see an experiment in which she, again, same protocol, distended the gut in control situations and got a small response. But when she put through the IBS fecal supernatant and then distended the gut, she got a much bigger response. And when we look at the summary of the high threshold units on the right, you can see that the supernatants from IBS patients actually increase the mechanosensitivity of these colonic afferent nerves. And we have some very recent data to suggest that protease may be involved in this, as well as histamine, and actually this also may be able to be modified by diet. But what about specific bacteria that might modulate pain signaling? Well, we can go back to the MET1 story, and what Jessica did here is she looked at specific bacteria, and when she took the supernatant from bacteria, Faecalibacterium praenutsii, it had a similar effect of the DRG neurons to increase the rheobase or lower the excitability, which again was blocked by the serine protease FUT175. She then looked at MET1, both with Faecalibacterium praenutsii as well as without it, and found that when the MET1 did not have Faecalibacterium praenutsii, the supernatant had no effect on the DRG neuron excitability. And this is the effect of this Faecalibacterium has also been shown in vivo, in which in this study, using a neonatal maternal separation model, which has been used as a model of visceral hypersensitivity, the Faecalibacterium perniciae was decreased in the NMS mice compared to non-handled mice. And similarly, in the bottom graph, you can see that the VMR response is increased in the neonatal maternally separated mice, but when they then gavage those same NMS mice with the Faecalibacterium perniciae, that visceral pain response was inhibited. When we then translate that, however, into patients, it gets a little more murky. As some studies have shown, for example, in IBS patients, that Faecalibacterium perniciae is decreased compared to controls, whereas other studies have shown there's no difference between IBS patients and healthy controls. And I think this not only reflects the heterogeneity of a disorder like IBS, but I think it also highlights what I said at the beginning, that the microbiota plays a significant role in abdominal pain in some people, but not everybody. And there are other analgesic microbes out there, just to sort of let people know it's not just Faecalibacterium, so lactobacillus can inhibit TRPV1 channels on DRG neurons, Bifidobacterium can produce GABA, which is inhibitory, and there's an E. coli strain which produces lipopeptide, which is an agonist of GABA B receptors. So in summary, while we know the host plays a significant role in secreting mediators that can modulate visceral pain, there's growing evidence that the microbiota also is an important modulator of pain, either indirectly by signaling to these host cells to release their mediators, or by directly interacting with the nerves themselves. So that the microbiota secretes both pro and anti-nociceptive mediators, and it's a teeter-totter balance between the anti-nociceptive and pro-nociceptive in terms of what the effect of visceral sensitivity, but it's unclear in which patients this balance is disrupted, so that we can really target in those people where it is disrupted for a microbiota therapy. And so in summary, I'd just like to acknowledge the collaborators and trainees who are involved in some of the work I showed you.
Hi. Thanks, David. That was an excellent talk and given a lot of food for thought for everyone to think about. We'll hold the questions till the end so we can move on to the next speaker, but please send your questions on the Q&A part of it. We'll move to our next speaker, Dr. Gerard Clarke. Dr. Clarke is a lecturer in the Department of Psychiatry and Neurobehavioral Science and a principal investigator in the APC Microbiome Ireland at University College Cork in Ireland. His current research focuses on translational study of the microbiome gut-brain axis in stress-related disorders like depression and IBS. And he has a particular interest in the role of tryptophan kynurenine pathway. He's also the past recipient of a research grant from ANMS, and today he will help separate fact from fiction when it comes to the role of microbiome in regulating gut-brain axis. Welcome, Gerard.
Thanks, Purna, and greetings from a very wet and windy Cork. It's a real pleasure to be part of this ANMS virtual symposia. So I suppose the title of the talk kind of refers to the fact that there's been a lot of promising preclinical and clinical research indicating that the gut microbiome might have a role in impacting brain function and behavior. And I suppose what people are starting to wonder now is whether these findings are going to have any clinical benefits. Are they the real deal, or are we looking at maybe a Trojan horse instead? So I'm going to try and provide some insights into that as we go along. So I suppose the idea behind this is that the microbiome or the microbiota hijacks the gut-brain axis, this bidirectional signaling pathway between the gut and the brain, and that this is a two-way system. So we know as well that stress, for example, perceived at the level of the central nervous system can also impact on gut function. And what the studies have shown us to date, as our first speaker has indicated, is that there's an important role for the microbiome based on these studies in pain, but also anxiety and stress. And we see similar observations in psychiatric disorders. And this is relevant, of course, then in neurogastroenterology, particularly in relation to disorders of the gut-brain axis like irritable bowel syndrome, where you have a constellation of these symptoms, including pain, a stress-related disorder, and also a lot of psychiatric comorbidity. So the approaches then that have been taken to date relate to a variety of different experimental strategies which have indicated the extent and scope to which the microbiota impacts on different behaviors. And a lot of these preclinical approaches are somewhat self-explanatory, but maybe the one that's worth exploring in a little bit more detail relates to the use of germ-free animals. And maybe because the germ-free approach in a way is a little bit of a blunt tool, maybe that's why the question that's, that forms part of the title of this talk has been posed, because these animals have been around for quite a long time, and it's only relatively recently, I suppose in the last decade or more, that they've been applied to answering the type of questions that we've been interested in terms of microbial regulation of brain function. But I suppose it's difficult to think of a real translational clinical equivalent where there's such a complete obliteration of the gut microbiota across the lifespan. But nevertheless, they're a really neat tool, and they can be used in a variety of ways to get a yes/no answer as to whether particular aspects of brain function might be impacted by the gut microbiota. So you can compare conventional animals to germ-free, and you can also do some manipulations post-weaning, for example, monoassociation studies or colonization of the animals to see if the particular aberrant phenotype that you've noticed as a consequence of growing up germ-free can be rescued at a later stage. Now, one of the main areas of host physiology that the microbiome has been implicated in on the basis of studies in germ-free animals relates to the stress response. And I guess we're all familiar with the current stressors associated with modern living and life at the minute. We have our own individual stresses on each side of the pond. We're still, even though it's fallen off the radar a little bit now, dealing with Brexit. We know as well that the current COVID-19 situation is a very stressful time. But I suppose it's not the stress that kills us, it's a reaction to it. And at least from a physiological perspective, the microbiota has a big role to play in controlling the stress response. So what we can see here are studies following on from the original observations by Pseudo and colleagues, where we've shown again that germ-free animals have an exaggerated stress response compared to control animals, as indicated by the production of corticosterone following an acute stress exposure. And that ties in pretty well with what we've seen from the administration of probiotics as well, where specific strains are able to reduce or blunt stress-induced corticosterone production. So I guess one of the ways that we have of trying to separate fact from fiction is trying to see whether these observations in the preclinical studies will also be relevant in clinical populations. And there are obviously some obstacles in moving studies from rodents to humans. A mouse is not a small rat, and neither are miniature humans. But nevertheless, and despite these challenges, there are some promising and good routes towards moving this from bench to bedside. So I just want to give you some examples where these translational attempts have been made, and this is one from our lab where we looked at this B. longum 1714 strain, which we screened preclinically, and it came up as a strain with potential benefits for stress and cognition. So when we administered this in healthy human subjects, we were able to replicate many of the observations that we've seen in our preclinical studies, including the blunting of the physiological stress response in terms of reducing cortisol output, and also in terms of having an impact on brain activity and improving aspects of cognitive performance. Now, our colleagues in Germany, Paul Inck, also used this strain in a different imaging study and with a different stressor, and also demonstrated that it had some benefits in modulating neural activity, and also having a beneficial impact on the stress response. So we were very encouraged by this. So we went back to try and see if we would see some similar benefits for another strain that we've worked on quite a lot here in Cork, which is the Lactobacillus rhamnosus JB1 strain. And despite the fact that this strain had one of the strongest preclinical profiles that we've seen, it did absolutely nothing when we moved it in to our healthy human subjects using a similar kind of protocol as the one that we had used for the biflongum. So I guess that illustrates that you know, the translational attempts are going to be very strain specific as well. And we still need to figure out why exactly one strain produced benefits and one didn't. Now, the other important aspect of trying to separate fact from fiction is trying to figure out precisely what the mechanisms are that could be underpinning these observations. And of course, if we look at signaling along the brain gut axis, there are many potential routes that could be at play. Now, one relates, as we've heard in our first speaker's talk as well, to the production of microbial metabolites like short-chain fatty acids, and the possibility that some of these short-chain fatty acids like acetate, propionate, or butyrate might be able to modulate brain gut axis signaling to have a beneficial impact on stress and anxiety. And maybe that's the reason where we have seen successful translation, why the microbes in question might be producing those particular benefits. So again, this is an animal study we did a couple of years ago, looking at the impact of short-chain fatty acid administration following a chronic psychosocial stress protocol. And just what I want to draw your attention to here is the fact that in addition to having a beneficial effect in the forced swim test, which is an indicative of an antidepressant effect, the short-chain fatty acid administration was able to blunt stress-induced corticosteroid production during the forced swim test, and you can see that here highlighted in the arrows. And then the reason I wanted to focus on this study in particular was because of this recent elegant study in humans from Lucas van Oudenhoeve and colleagues, where they took a very neat approach in delivering short-chain fatty acids to the colon using a formulation-based strategy. And again, we're able to show in humans that what has been previously demonstrated in the animal studies, that the short-chain fatty acids were able to blunt the cortisol response to a psychosocial stressor. Now, of course, if we think how this relates to neurogastroenterology and disorders of the gut-brain axis like irritable bowel syndrome, where there's thought to be a heavy role for the gut microbiome now, and we can think about some earlier studies that we've done in irritable bowel syndrome, looking at the acute stress response, whereby subjects with irritable bowel syndrome tend to have an exaggerated cortisol production during a stressor like this. And then we can think maybe that this is a pathological situation where we might be able to test further the translational impact that we've seen thus far. And I guess that's one of the areas that's lagging a little bit behind, and that there's a lot of studies in the preclinical environment. There's a reasonable amount of studies now as well, looking in healthy human subjects, and we really need to see more interventions targeting the gut microbiota in particular pathologies. Now, another area, I suppose, that's been of interest to us is whether there might be an alteration in the gut microbiota in psychiatric disorders like depression. And after a number of smaller studies, this recent one from the Jerome Reyes group showed in one of the largest populations to date, and they also had a validation cohort in this study, that the initial indications that there might be alterations in the gut microbiota composition in depression seem to hold up in these larger studies. Now, there's also some efforts to try and see whether the microbiota might have a causal role and whether there might be a mechanistic basis for that. And I suppose there's an overlap there in terms of the neurobiology of depression being associated with alterations in tryptophan metabolism, while in parallel studies, including from the labs here in Cork, showing that there's a big role using these preclinical approaches like the germ-free animals for microbial regulation of tryptophan metabolism. Now, one of the ways in the field of demonstrating causality is the transfer of the disease-associated microbiota. So in this case, from our earlier study, we were able to show the transfer of the depressive phenotype by the transfer of the depression-associated microbiota, particularly in terms of anhedonia-like behaviors, anxiety-like behaviors, but also the classical neurobiological phenotype of depression in terms of alterations in tryptophan metabolism. Now we're attempting as well to try and get some mechanistic insight into this. So I'll just mention briefly that often when we talk about the gut-brain axis, we tend to omit the liver, and that may be an important site mediating the effects of microbes. And indeed, we've seen evidence that some of the enzymes in the liver, which are important in tryptophan metabolism, are expressed at lower levels in our germ-free animals, which would be consistent with the observation of increased availability of tryptophan. Now, it's also important to point out that this is not a static system, and that we know that stress alters the gut-brain axis signaling, and we've recently shown that it alters serotonergic signaling just following an acute stressor on the gut-brain axis, and that it does so in both a region and sex specific manner, which is contingent on the microbiota. And this is work we've done recently with some colleagues in the Air Force Research Laboratory in Dayton, Ohio. And let's give a shout out to Nancy Kelly-Nutnan and Michael Goodson there. So I suppose what we want to get to eventually is the possibility of therapeutic targeting of the gut microbiota with these psychobiotic options. And our current thinking really is that if we can get a better handle on the mechanisms that might be available via specific microbial interventions, then we can pair that up with the physiological alterations or the neurobiological alterations that are present in particular brain-gut axis disorders. So just to summarize and conclude, I suppose we've seen a lot of promising preclinical and clinical research that indicates the microbiome regulates behaviors in physiology, which is very important in neurogastroenterology. We need to think about increasing translational efforts. I've given you some examples where that has been both successful and unsuccessful. And it's important to move on to some mechanistic insights, focus more on causation, and then that might help us further establish whether this is fact or fiction. But I would point out that we should expect some attrition maybe along the way, and there may not be perfect translation on our quest to develop microbial-based strategies for the treatment of stress-related gut-brain axis disorders. So I'll just finish up there with some acknowledgements, and thanks for your time and attention.
Thank you, Gerard. That was an outstanding talk, and brings up a lot of questions. Again, we'll save them for the end of the talk. Please send your questions on the Q&A, and we can now move on to our last speaker, Dr. Premsyl Bercik. Dr. Bercik is a professor of medicine at McMaster University and consultant gastroenterologist at Hamilton Health Sciences in Hamilton, Ontario. He's also the Richard Hunt AstraZeneca Chair in Gastroenterology at McMaster University. His research focuses on the role of intestinal bacteria and gut-brain axis signaling and its relevance to functional GI disorders and gluten-related disorders. Today, he'll educate us as to how diet can be used to modulate the microbiome gut-brain axis. Welcome, Premsyl, and we look forward to it.
Thank you, Purna, for the introduction, and for those who wonder how I appeared on the program. So I'm filling actually for my colleague, Dr. Giada De Palma, as there was an unexpected twist in her pregnancy, and baby boy Oliver has arrived a few weeks earlier. Anyway, I was given this topic, which I think would deserve a whole day symposium, but and can be difficult to cover in 15 minutes. However, I will try to give some overview and maybe some examples, especially show some new data. So I would like to start showing that diet is one of the main determinants of microbiota composition and also function. And this is one of the first seminal studies from Gary Wu, who studied how early short or long-term diet affects microbiota composition in healthy volunteers. And in this graph, we can see that the sudden changes in diet make some shifts in the microbiota composition in individuals, but overall there are still differences between subjects participating in the study. However, when you look at the long-term dietary habits of these individuals, we can see that there are two different enterotypes, and they are mainly linked to whether these patients are mainly meat consumers or they are more focused on the vegetarian diet. the role of microbiome diet has also been examined in other studies. And again, I would like to show the very first one from, this is a group in Cork, which demonstrated in a relatively large cohort of elderly subjects that gut microbiota composition correlates very well with the diet, but also with some health or disease parameters. And although this is a very complex picture, what it shows that those individuals who stay at home and have a relatively diverse diet have different microbiota composition and appear healthier compared to those patients who are either in long-term care facilities, in hospitals, and also have a different type of diet. And in those individuals, the microbiota and diet correlated with increased inflammatory markers, decreased functional independence, altered mini-mental status, and also increased depression rates. So during my talk, I will be mainly focusing on irritable bowel syndrome because it's a kind of prototype of gut-brain disorders. And we know that both the diet and microbiota play a key role in this disorder. With respect to the symptoms, it has been reported by many studies that patients with IBS feel that their symptoms worsen when they ingest certain foods, and that on the contrary, specific diets alleviate their symptoms. And one of the diets which is very popular is FODMAP. So these are fermentable oligodemonosaccharide and polyols. These are poorly absorbed carbohydrates, and they have been linked to genesis of GI symptoms. Now, when we think about the possible mechanisms, so first, the FODMAPs, it's a quite heterogeneous group of compounds. So we have fructans, we have galacto-oligosaccharides, we have lactose, and it has been clearly shown that actually lactose intolerance can trigger symptoms kind of similar of IBS. And we have also fructose and polyols, which actually polyols especially can be used as laxatives. And apart from the osmotic overload, which these substances will trigger when they arrive in the colon, they will also be associated with bacterial fermentation and possible dysbiosis. And this can lead to gas production, genesis of bioactive metabolites, and also some immune stimulation, for example, through production of LPS. Now, what do we know about the effect of low FODMAP diet? So this is the result of meta-analysis which was done by a group of Paul Moyeri at McMaster, which reviewed the effect of low FODMAP diet in IBS. And as there is an overall benefit in patients with IBS by reducing these low fermentable carbohydrates, if you look at these three studies which are now highlighted in red, these are the studies which were the most rigorous design, and they were also least heterogeneous. And however, the magnitude of the effect of withdrawing FODMAP or reducing FODMAP was the least from all these studies. So several studies also noticed that responsiveness to a low FODMAP diet may be predicted by fecal microbial profiles, and actually our group in collaboration with a group at Queen's University studies this, and we think that the production of histamine by bacteria can be responsible for that. Another dietary component which is quite discussed recently is wheat or wheat sensitivity, and there are again several mechanisms of how the symptoms actually can be generated. And one of them is directly by gluten peptides, which can act through, for example, PAR2 receptors. And I want to stress that actually this mechanism of action is different from those which are observed in patients with celiac disease. So in gluten sensitivity or gluten sensitive IBS, we think that the gluten peptides can be acting through PAR2 and stimulate the immune system. But there were several studies recently showing that, for example, Pseudomonas can modify the gluten peptides, cleave them, and actually magnify the immune responses in patients containing these microbes. ATIs is another component of wheat, and this stands for amylase trypsin inhibitors. And again, these are compounds which are known to stimulate TLR4s, against important part of the innate immune system. And again, studies in Elena Verdu's lab showed that lactobacilli seem to be interacting with ATIs and possibly decreasing their immunostimulatory effects. So what do we know about the effect of gluten-free diet in IBS? So again, going back to the same meta-analysis which I showed earlier, there were only two studies, two RCTs, which examined the effect of gluten-free diet in IBS. And overall, the meta-analysis was negative. So there seemed to be some effect, but overall, there is, I would say, studies are missing. So we have actually examined this this question in a study which was just accepted in clinical gastroenterology and hepatology, and we studied actually effect of 1 month of gluten-free diet in patients with IBS, defined by ROM3 criteria, and healthy subjects. And we enrolled 50 patients, 25 healthy volunteers, and we studied their IBS symptoms, but also psychiatric comorbidities, intestinal transit, before and at the end of this 1 month of gluten-free diet. And these are the results. What we found that when we stratified the patients based on presence of anti-gliadin antibodies, and specifically IgG antibodies, we found that those patients who had these anti-gliadin antibodies were responding more to the gluten-free diet. So in this group, it would be 75% had significant improvement, compared to 38% in patients who had who were without anti-gliadin IgG antibodies. And again, this goes very well with the placebo effect between 30 to 40%. On the right, you can see the magnitude of the improvement, and looking specifically at change in Birmingham IBS score. And again, the magnitude of the effect is much more pronounced in patients with anti-gliadin IgGs. When looking at the factors which could actually predict the improvement, and we actually created model where we place several factors, including, for example, genetic susceptibility to celiac disease, adherence to the diet, improvement in the gastrointestinal transit. We found that it was actually the presence of the IgG antibodies, and surprisingly, the adjusted OR was almost 130. So there is the conclusion from this study that gluten withdrawal may actually improve symptoms of IBS and also the psychiatric comorbidities, at least in this group of patients. We have, as a part of our diet, we consider them, prebiotics are part of our diet, right? They are sold as dietary supplements. And I wouldn't be going now to the IBS itself, because there were several meta-analyses, including the recent technical review and recommendations from American Gastroenterology Association with the use of probiotics. But I would like just to share with you the effect of, sorry, I'm speaking about, I should be speaking about prebiotics. So I apologize. This is the effect of fiber on psychiatric comorbidities, especially depression and anxiety. And what this recent meta-analysis from Liu and colleagues published last year showed that prebiotics or administration of prebiotics does not improve depression or anxiety, as shown in these two graphics. as you saw, I mean, there were only several studies which were actually studying this, this topic. And I would like to highlight that actually we have, significant amount of data in animal studies, which would suggest that probiotics can affect both behavior and also stress responses. And specifically, this is actually study again from one of the previous speakers, a group from Cork, when mice were subjected to chronic social stress, and they were either administered FOS, I mean fructooligosaccharides, or galactooligosaccharides alone or in combination. And from the graphs on the left and in the middle, you can see that the administration of this prebiotic was able to decrease the anxiety and depression-like behavior. And on the right, you can see that even stress responses measured by the corticosterone levels were decreased by the administration of prebiotics. So I think that we need more clinical data to really make the proper conclusion whether the prebiotics could be used therapeutically for psychiatric comorbidities. Now, with respect to probiotics, and again, this is, as I said, this is a review which was focusing on symptoms of anxiety and depression. There are a long list of studies that, and the overall conclusion is that probiotics improve symptoms of depression. What I would like to caution that some of these studies actually use the symptoms of depression as a secondary outcome. Some of them, when looking at the raw data, were not really providing the information about the baseline status of depression or anxiety. However, if you look at some of these studies, there seem to be benefit, at least, of certain probiotics in depression. We did actually a study of our trial is part of this meta-analysis where we studied effect of Bifidobacterium longum in anxiety. And you can see that after 6 weeks of treatment, this probiotic improved symptoms of depression, and the beneficial effects were present even 1 month after. This was associated with changes in overall IBS symptoms, and when looking at the brain, because we performed functional brain imaging, we could see that the treatment with this probiotic decreased activation in the amygdala region, which was the primary region of interest, an area which is actually involved in controlling depression and anxiety. And when studying also the other areas of the brain, we discovered that Bifidobacterium longum caused a lesser engagement of the frontal and temporal cortex compared to placebo. Again, these are the areas which are involved in control of emotions. There are multiple ways through which the bacteria can affect the brain, and actually we think that in the case of B. longum, it can be the interaction of the noradrenaline to dopamine conversion in the host. However, at the end, I mean, I have just 2 more slides to show that we could maybe look not only in the bacteria, but also in other components of the microbiome, and this is specifically fungi. And this was a study which was, again, just accepted in neurogastroenterology and motility. In this study, we used a model of humanized mice colonizing germ-free mice from microbiota from either a healthy individual or a patient with irritable bowel syndrome with comorbid anxiety. And the second group was actually given either water alone, or they were given Saccharomyces boulardii, which is a known probiotic used commercially. And we saw that colonizing mice with this IBS microbiota increased gastrointestinal transit, caused also anxiety-like behavior, and these abnormalities were corrected actually by administration of this fungus. When looking at the mechanisms, we noticed that within the microbiome profile, there was an increase of the genes which were involved in indoor biosynthesis. And these indoors are actually one of the modulators of AHR activity. And when looking specifically at the production of indole 3 acetic acid, which is a known agonist of aryl hydrocarbon receptors, we saw that there was a clear increase in this group. So this is just my take-home message. So long-term diet is the main determinant of the microbiome composition and function, and it has been linked both to human health and disease. Diet seems to play a major role in IBS, and we know that gut microbiota may modulate many effects of many of the dietary components, whether it's FODMAPs or gluten. We have so far clinical data that suggests that probiotics, but probably not prebiotics, alter symptoms of anxiety and or depression. And it seems that there are multiple pathways by which diet can impact this microbiome gut-brain signaling, and whether it's the short-chain fatty acids, which I already mentioned earlier, immunomodulation, or production of the neuroactive molecules. And I would like just to acknowledge all the collaborations and the fundings. Thank you.
Thanks, Premsyl. That was brilliant. And again, I think I would like to thank all the speakers for taking time out of their busy schedules in such a time to come share these findings and educate us on this important topic. I think we can move to the discussion. I saw a lot of interesting questions pop up while the talks were going on, so I'm just going to get right to them. I think the first few were for Dr. David Reed, but anybody can go ahead and answer them. The first one from Dr. Rosa Uribe and Rice University raised an important question of why would microbiota modulate pain? In fact, what's in it for them by doing that to the host? And just in the same breath, Dr. Bolemba from University of Idaho was asking about more specificity in terms of what proteases influence nociception.
I guess because I did the pain talk, I'll start. You know, it's a good question. What's in it for the microbiota in terms of why they would modulate pain? But I think, so that's a more difficult one to answer. I guess one of the highlights of the work with the MET1 story is that, in fact, we often think about the microbiota being pronociceptive in disorders, but in fact, there may be this sort of inhibitory tone provided by the microbiota in health. And that it's maybe simply we lose that inhibitory tone in certain disorders rather than gain, and then that may then swing the balance over to sort of more pronociceptive signaling from the microbiota. In terms of the proteases that are involved, that's a very good question, because you know what I showed there with the MET1 story is serine protease acting through part 4, but we also know serine proteases also can activate part 2, which on DRG neurons will be cause their excitability to increase. And so there may be specific, we don't know the specific proteases that are involved, but there may be specific ones that are acting through specific receptors, or there may be other mechanisms whereby the balance between what's secreted by the microbiota is also influenced at the level of the neuron, for example, for internalization of receptors, which may have longer signaling via one receptor and very sort of short acting signaling via another receptor.
All right, thanks David. I'll follow up with the question from Dr. Pandolfino from Northwestern. He had a more general question for the entire panel. So the microbiome alterations and their effect on the colon, is it specific to the colon, or you also see the similar effect in other parts of the gut? Of course, he's more interested in the esophagus to see if similar effects have been demonstrated in the esophagus, but I'd say even the small intestine is fair game.
So if I may start, actually, at least in animal models, we actually see more effect in the small intestine than in the colon.
But not in the esophagus.
Not in the esophagus, sorry. I haven't studied it yet.
All right. Did anybody else want to add to that or no? Just highlights that, you know, it's surprising that, you know, all the work has been done in the colon and it's sort of people have forgotten about the foregut. So I think it'll be interesting to see more data come out in this space. I'll follow up with a few more questions. Dr. Suez had asked if adolescence is a period of vulnerability and maybe carries a different microbiota from adults. So would stress, diet, and other modulators have more profound effects on the microbiota and the neurophysiology and subsequent behaviors during this period? Or if you were to think of it differently, does the stage of life matter in terms of perturbations that can happen in the microbiome in terms of long-term effects? And any one of you guys can feel free to jump in.
Yeah, maybe I'll jump in there, Purna. You know, I think the idea of critical windows is pretty important, both in terms of the vulnerability of the microbiota and also the vulnerability of the central nervous system. Now, there's probably been less done in adolescence rather than earlier life stages. So we know, for example, that early life stress, the maternal separation model, leads in adulthood to a phenotype which is very reminiscent of IBS. And these effects may be mediated via the gut microbiota because the imprint of that early life stressor is still visible in the microbiota even in adulthood. Now, other work from Shimona Mahoney here in UCC has kind of shown that you may even have effects that are clinically silent initially, which may subsequently manifest in adulthood as alterations in abdominal pain, for example. So in a study using early life antibiotic administration, she would have shown that even though the effects of the antibiotic had resolved in terms of the footprint on the microbiota, that the visceral pain phenotype still persisted in adulthood. So I think this concept of critical windows is very important, and adolescence is probably one of those windows where we need to look at a bit more closely.
And just on a related note, Dr. Srinivasan from Emory University has asked how short-chain fatty acids can lead to reduced cortisol levels and regulate anxiety and stress, and more generically, where are they? What level are they acting at? Yeah,
so I don't think it's fully clear yet where exactly the short-chain fatty acids from the studies that I discussed are having their effects, and there is some question over which of the particular short-chain fatty acids in the mixture that was used would actually reach the central nervous system and potentially signal there. I guess acetate, if we were going to put a bet on it, would probably be the one which would be most implicated. But it's possible as well that the short-chain fatty acids are signaling via other mechanisms locally that subsequently kind of get transduced to the central nervous system and then feed in from different relay centers to the brain regions which are important for the control of stress and anxiety. So the jury is out on the level at which that's happening just now.
Great. Thanks, Gerard. The next two sort of related questions are for Premik. The participants with anti-gliadin IgG antibodies who responded to gluten-free diet, did they belong to a particular IBS subtype? And do you suspect there's a unique pathophysiology to consider the interaction between microbiota and these IBS subtypes? And on a related note, should anti-gliadin antibodies be used for inclusion or exclusion studies for assessing IBS patients?
Well, so first I will start with the second one. I think that it's part of the IBS. I mean, we try now to understand what is actually triggering the symptoms. So we have another ongoing clinical study to look for specifically some, whether it's gluten, whether it's ATI, whether it's inulin. For me, it's just that IBS is this big black box, you know, and I think that we should look for the specific mechanisms. And I think that the anti-gliadin antibodies is a marker for us that the wheat is involved in the pathophysiology. So it would be just one of the underlying mechanisms. Otherwise, with respect to the type of IBS, we saw all four subtypes of IBS actually in patients who had these IgG antibodies. What is very interesting that the great majority had IgG. We haven't seen basically IgAs, which are, I think, more aligned with celiac disease.
Right. Well, we still have you online, Premsyl. There's a question on the guidelines you just wrote for AGA that probiotics do not help symptoms of IBS, and they're asking if you have any comments.
Well, I would like to point out that actually Purna was part of the technical committee. So I think that this is actually because we don't have enough clinical data. There are several clinical studies which suggest that the probiotics may have beneficial effect, but they differ in methodology, differ in the types of probiotic used. And I think that what came from these guidelines and from the technical review, what is important that we have similar approach which we see in commercial therapeutics, I mean commercial medication. So we should have really big studies, well designed, which will tell us whether that specific probiotic is beneficial or not.
Great, thanks, Premsyl. I know we are over time, but we have a few more questions. So if you guys are willing to stick on, I can try to cover some of these while we're here. One of the questions was about curcumin and if there is any data on curcumin and depression and changes in the microbiota. I'm not aware, but if you guys know of any, feel free to chime in.
I mean, not to my knowledge. I know that curcumin has anti-inflammatory effects, so whether this would be the mechanisms of improved symptoms, but I haven't found any studies on that.
And are there any studies on diet and the gut-brain axis in children?
I must admit that I'm not sure, actually. I always look more for the adult patients.
It's a good reminder to us to look at more of the pediatric data. Gerard, do you know of any studies?
Not that I can bring to mind at the minute. I mean, there's a lot of interest actually in modulating the maternal diet now to try and see whether we can set the maternal microbiota on the right course for subsequent transmission to the infant. But there's probably less direct intervention studies that I'm aware of in that age group.
Good. David, this is specifically for you. Would experiments in epithelial-specific PAR4 knockout mice demonstrate that this is direct communication between PAR4 on neurons in vivo?
That may be, I think, a way to sort of try and sort that out. If we could knock out the specific receptors and specific cell types, I think then we can get into the more indirect versus direct components. So that would be, I think, a really neat way to go at it. Yeah.
So the other two are sort of related questions. I'll try to build them together. If there's been a mouse model of SIBO or small intestinal bacterial overgrowth to look at in terms of nociception, and what regions of the intestines are good to study neurotransmitters and why?
Well, maybe in terms of the last question, I suppose it depends on the context. I mean, there's an abundance in neurotransmitters, particularly serotonin, very abundant in the gastrointestinal tract and widely distributed along the biogeography of it. I suppose even to the fact that it's present in the lumen and can be liberated by certain bacterial enzymes, and we probably have less of an appreciation of what the specific role of free serotonin in the lumen is, other than it may be a stimulus for the bacteria themselves. So there's a lot to that question. It really depends on the context in which you're interested, I suppose.
And I'm not aware of a mouse model of SIBO, or if somebody's looked at that. Premsyl or David, do you guys know anything?
No.
And so I'll end with the last questions. What is known about sex hormones and microbiota? I mean, there's described a difference in the gut microbiota between men and women in terms of diversity and composition, but I don't know of direct data. Of course, I'm sure that exists, but I don't think I know if it, any of the panelists know about data around that?
I mean, we've seen sex-specific differences in a lot of the readouts, with a lot of the effects on brain and behavior, for example, more prominent in male animals. But we don't know specifically why that is yet. The obvious thing to look at would be in terms of ovarian hormones, but that still has to be teased out a little bit further, I think, before we can be too definitive on it.
Great. That was a really exciting session and really interesting questions. So I want to thank again the panelists for staying longer, and then also for all the attendees for taking time out of the day to come join us. Thank you all. It was great seeing you all again.
Thank you.
Bye. Thanks.
Bye-bye.
Thanks, everyone.
Yeah.
Thank you.